Greetings everyone, and welcome to another exciting new genetic engineering and biotechnology news webinar. Our presentation today is entitled Genotyping by Sequencing using Single Primer Enrichment Technology, or SPET. I'm Jeff Bogaliskis, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Next-generation sequencing technology continues to dominate genomic sciences, driving sequencing costs down to levels where the sensitive technique is becoming a practical solution for all types of high-throughput studies, such as genotyping by sequencing. However, addressing genomic complexity still remains a challenge when performing targeted sequence analysis, and companies are developing tools to tackle issues currently facing genome investigators. Let's meet our panelists for this GEN webinar, who will discuss the novel approaches for targeted sequencing of DNA that is suitable for a wide range of genomic targets. Our first speaker today will be Luke Sherlin, Director of Technical Support at IGA Technology. Dr. Sherlin will introduce us to Nugen's Ovation Target Enrichment System and how it can be applied to create a more flexible and accurate laboratory workflow for various gene samples. Our second presentation will be given by David Scaglione, Senior Research Scientist at IGA Technology Services in Udine, Italy. Dr. Scaglione will describe how he was able to utilize single primer enrichment technology to rapidly and accurately genotype a massive number of samples for genome-wide association studies. So before our speakers get started, I want to encourage the audience to submit questions for our Q&A session at the end of the presentations. We will try and answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. So let's begin our webinar and open the floor to our first speaker. Luke?
Greetings, and thank you to the organizers for the opportunity to showcase a unique application that is being done using the Nugen Ovation Target Enrichment System. Targeted resequencing is currently used to reduce sequencing costs by focusing on specific regions or events of interest within a genome. While the costs of sequencing do continue to go down, the costs associated with whole genome or whole transcriptome approaches in order to capture all the relevant information are still untenable for the vast majority of researchers. At the same time, it can be costly in terms of price, time, and sample to invest in multiple assays to approach all the different questions to be answered by the same DNA or RNA sequence. An important application that requires cost-effective sequencing is genotyping, which you can see at the top of the applications wheel. Particularly in the realm of agrogenomics, a large number of sites need to be interrogated quickly and in a cost-effective, high-throughput fashion. This is the application we will be hearing about in detail today. The Nugen Ovation Target Enrichment System makes available a single low-input workflow that is applicable to a variety of different workflows and applications. In addition to genotyping, the system can also be applied to interrogation of cancer-related aberrations, including mutations, CNVs, and gene fusions. Other disease-related applications include viral integration mapping, pathogen characterization, or identification of germline aberrations. Fundamental research can also be facilitated in discovery of gene fusions or focused measurement of gene expression and alternative splicing. These are just a few of the many applications that can be addressed within a single workflow and often within a single assay and sequencing run. Today the focus will be on how the Ovation Target Enrichment System can be applied as an efficient system for genotyping. As we will see, the system offers a number of unique benefits for a cost-effective, accurate, high-throughput method to genotype virtually any organism. It is clear that a specific application requires more than an efficient library preparation platform, but it also requires a robust workflow solution in order to obtain a valuable readout. We will learn shortly about the details of a genotyping workflow that has been developed in concert with our colleagues at IGA Technology. What you see here is another example of a workflow as applied to cancer research. As you can see, the sequencing and sample preparation, while important, are only a part of the overall workflow. Also key to a successful application include procurement and storage of samples and sample isolation and characterization. It is vital to work closely to ensure that the samples are isolated and stored in a consistent manner. An example of this is Nugen's work with the RUCDR Infinite Biologics and Biostorage Technologies at Rutgers, where we have validated an end-to-end RNA-seq workflow from whole blood samples. Getting your samples on the appropriate platform requires library preparation, which in some instances can be a bottleneck. These can be greatly improved with a flexible, low-input preparation solution such as Nugen's Ovation target enrichment systems. After data acquisition on various platforms, it is critical to have a robust analysis pipeline that will include relevant readout so that the data does not become the bottleneck. This is also a focus, and Nugen is providing solutions in a number of different ways in order to ensure this can be done quickly and easily. As an example of this, Nugen approaches data analysis in a manner that provides the most flexibility, ease, and transparency to a researcher. By providing tools that exist on the sequencing platform through the use of Illumina's base space, researchers are able to get valuable readout on samples with just a few clicks and without bioinformatics expertise. For researchers that have greater expertise and develop their own pipelines and software tools, software programs and scripts for processing and analysis are readily available, along with a support team to help ensure the readout is informative. These workflow solutions have already been applied to Nugen's Ovation target enrichment solutions. The flagship Ovation DNA Cancer Panel and RNA Fusion Cancer Panels provide robust discovery tools for cancer researchers. These panels can be expanded, or target regions can be focused in custom panels. Customization extends beyond applications in cancer to agrigenomics applications, which we will hear about shortly, and extends to exploration of any aberration in any known genome. This slide provides a snapshot of a few different custom designs that have been successful with custom solutions. As you can see, there's a broad range of target sizes from a single exon to hundreds to thousands of genes. The table also shows a variety of different species, including plants, mammals, and microorganisms, to different sample types, and includes a variety of applications that can be explored with fast and flexible customization. Today's focus will be on genotyping for agrigenomics. There are an abundance of additional applications in this space that can be enabled with Nugen's target enrichment system, including large-scale rearrangements, pathogen mapping and identification, and fundamental research, including gene expression. At this time, I will hand off the presentation to Dr. Davide Scaglione so that you can learn more about the workflow they have developed with the target enrichment system.
Thanks, Lou. That was a great presentation to kick off our webinar. I think you showed the audience not only the versatility of the Ovation system, but also the numerous potential applications where the technology could help streamline next-generation sequencing workflows. Before we move on to David's presentation, I want to remind everyone once again to submit questions for our Q&A session at the end of the presentation. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All of our speakers will be available to answer your questions after the final presentation. So let's get our last presentation underway. David, the floor is yours.
Hi, everybody. I'm Davide Scaglione, and I work as a bioinformatics researcher at IGA Technology. With this webinar, I would like to show how we use the SPET technology from Nugen to pursue a large-scale genotyping-by-sequencing experiment. In the last years, array-based solutions have dominated the market of genotyping, and they did provide very robust assays. However, with the synthesis cost of probes, they used to require a very high investment at the beginning of projects. And also, they suffer some constraint in the minimum order of samples and assays. So this provided a limited flexibility in the throughput. And also, mainly, they suffered the so-called ascertainment bias problem, which means that researcher used to sample the diversity that was used to design the panel instead of discovering the novel diversity within the given sample under the research given to be plant germ plasma or animal diversity. On the other side, next generation sequencing technologies have posed on the market with a keep decreasing cost, also providing a greater scalability given by the capability of multiplexing reads. They also do provide sequence information, and so they are also potentially an ascertainment bias-free technique. The technological improvement of NGS systems, in particular, that one of Illumina, have seen a significant increase in read length. This has also facilitated the publication of many draft genomes. However, some challenges still persist when it comes to optimize genotyping-by-sequencing techniques. The most important is the ability to fit with the complexity of a genome under analysis. Then it comes the multiplexing capability, which play a major role in for the ease of scaling up and down with the number of desired polymorphic markers. And all of this must be coupled with a good cross-sample coverage to ensure a very high genotyping ratio. It's also worth mentioning that some approaches of genotyping by sequencing rely on a very low coverage per locus, sometimes leveraging on the ability to trustfully genotype inbred lines with a single haplotypes or the ability also to perform some imputation. But however, this is not the case when dealing with the heterozygous or even polyploid genomes, where the imputation is not so easy and some prior information will provide cannot provide robust imputation capability. Some well-known examples of complexity reduction are restriction enzyme-based, the so-called RAD, restriction-associated DNA, or GBS. Some protocols relying on the sequencing of every fragment involved in a restriction cut. Others instead are relying on two enzymes and library fragment selection, which can provide a more precise and dedicated complexity reduction. Depending on the type of analysis, different amounts of data points are required to obtain the adequate resolution. For instance, a limited amount of data points are necessary to resolve the structure of population, while for linkage analysis, some thousands of polymorphisms are necessary to saturate all possible recombinations. So moving up to association studies, the linkage disequilibrium decay is driving the maximum span of two markers to maintain the statistical power and the ability to successfully detect association to a trait. Therefore, we can say that the perfect versatile genotyping system is the one that can easily scale across all these scenarios while maintaining its cost effectiveness. In our case study, the goal was to conduct a genome-wide association study in Populus nigra. However, given its rapid decay in linkage disequilibrium, the possibility of utilizing a random reduction of the genome, such as that provided by the previously mentioned restriction enzyme systems, was not preferable solution, as it would generate data points with a very low likelihood to be associated to some phenotypic traits. Instead, we aim to have a genotyping method to specifically target the gene space, but not all of it, which would be prohibitive in sequencing cost term. Only some specific loci or SNPs were our target. And the available resources that we can leverage were the genome assembly of Populus trichocarpa and a low-pass sequencing of 54 different accessions of Populus nigra, and also some previously genotyping data coming from the Illumina Golden Gate platform, which is an array-based platform. We decided to consider the SPET technology from NuGen. This technology utilizes a single primer enrichment system to enrich a short fragment library for target region. In its original design, one end of any fragment is defined by the annealing site, which determines the enrichment for a particular locus, while the others, being generated by fragmentation, are scattered across a region of some 200 to 300 base pair. And they are the only ends that are going to be sequenced in the original system, looking for unknown or causative polymorphism, or in other cases for structural variations. The typical structure of the library is depicted in this slide, where the right end is defined by the probing site. The other, generated by sonication, contains the sample barcode along with a sequence of 6 bases, of 6 random bases, which are used in downstream analysis for removing the duplicates of PCR. However, given our purposes, the idea was to adopt this technology, but with the maximization of the genotyping ratio for a set of chosen SNP sites of interest. So instead of targeting a region with the distributed coverage of reads just by means of the first read of a pair, we managed to design probes to be just upstream of a target SNP in order to be read through by the reverse read. So in this way, the maximum distance is given by the reminders of the sequencing length after the probe itself. One caveat was to avoid at most any further SNP site to reside in the 3' end of probes and avoid any allelic specific extension. However, we also sequence, as in the standard approach, the forward read, which can always provide the discovery and genotyping of other off-panel SNP sites. This approach can therefore provide a targeted genotyping solution, as we used to know it in the Arraybased platform, with some unprecedented benefits, which is the ability to discover novel diversity and also to filter out duplicate reads, which is something that was missing in the original, in the standard complexity reduction approach, such as RAD or GBS. So we choose the list of SNPs by prioritizing their distance to coding regions of the genome, so moving from exon to UTR region and then up and downstream regulatory regions. We also scale the number of SNPs depending on the gene length, and also imposing a minimum distance between SNPs residing on the same gene. Also, we applied a cutoff for the minor allele frequency. And we proceed after designing the full panel of 110,000 SNPs. We started by synthesizing a first test panel of 12,000 probes, which was assayed on 8 samples, producing some 1 to 2 million pairs per sample. This plot showed the distribution of allele frequencies as we did select from the low-pass sequencing data that we had in our hands. So just keep in mind this profile. So with the first data back from our lab, we assessed the percentage of enriched bases, or in other words, how many reverse reads aligned on the probe regions original designed. Data showed that this enrichment system, which here was applied on an interspecific context of Populus nigra based on Populus trichocarpa genome, outperformed standard hybridization-based systems we have worked with so far. Even when using curated probe sets in model organisms such human, we never saw such a very high percentage of enriched bases. To assess the cost effectiveness of the enrichment and the sequencing, we analyzed the distribution of coverages on target SNP sites. Distribution are kind of wide, as you can see in the left box. This is also expected in a single primer extension method, where small differences in the annealing efficiency can have a dramatic impact in the final amplification product. Since most SNP sites reside below the threshold of 200x coverage, we did not report over-amplification issues caused by repetitive regions or such. Moreover, if we look at the correlation between samples, we can notice that low and high-performing probes are reproducible across experiments. Therefore, drops in the calling efficiency will always and only impact certain subpopulation of probes, maintaining a high calling ratio for most of the others. So, this means that there will be never a combinatorial effect of calling efficiencies which will generate a drop in the genotyping ratio efficiency. Moreover, it must also be said that NuGen support the custom redesign of some particular probes that are poorly performing. So a user can always go back to the original design panel and adjust the probes based on their efficiency. We also need to note that this was a first-run greedy experiment, which means that we wanted to squeeze as the most the system by just using a single probe per SNP site, which was not the original idea of this system. So to assess the ability to get a smoother coverage by using more than one probe per SNP site, we analyzed some data that we generated for another experiment in Vitis vinifera, where we made up to 4 probes per SNP site. In these plots, you can see how moving from 1 probe per SNP site to 2 probes per SNP site dramatically reduced the amount of zero coverage loci. And also, if you look at the red plot, the green plot, you can see how the distribution curve approach a normal distribution. So we can see that statistically there is a benefit in using more than 1 probe per SNP site. However, I must comment on this that there is not an adequate payoff on doing such as to synthesize the double or to 2 or more or 3 times the probe. We just get a few more robust data points in front of a higher cost of probe synthesis. Also, by retrieving some previous genotyping data from a golden gate assay, we managed to compare calls for some 100 SNP sites in other 6 samples of the test panel. As the plots show, the coverage is the only limiting factor to a 100% accurate call. The system itself does not introduce a significant bias in allele calls. At lower coverages, the only error are false homozygous call or missing data. With these metrics in our hands, we simulated the error rates in different sequencing setup, foreseeing the 90,000 probe panel. So the need to maintain the high cost effectiveness on a large scale, we decided to consider the sequencing of 48 samples per Illumina lane. And with this system, we simulated that the error rate for both homozygous, false homozygous call and missing data would be around 1.5% each. So the final sequencing setup would generate some 8 million reads per individual on a 90,000 loci panel and for a total set of 480 samples. So we have now completed the sequencing of 479 samples with the 90,000 panel. And as you can see in the sequencing yield across sample is well distributed, but for some 20 samples which suffered poor sequencing coverage, which was basically due to the poor quality of the input DNA. And also we managed to obtain such an even distribution of coverage, not going through a normalization step of DNA, but another procedure I'm going to show in the next slide. Instead of performing DNA normalization, we operated a rank-based resorting of DNA after the sonication step. And then those ranks to be pooled for the enrichment procedure. This ensures that samples within a pool are homogeneous in quantity. Also, on our experience on such big population where DNA is extracted from different substrates in different conditions, the final DNA yield is also a proxy of its quality. So therefore, pulling together samples with a similar DNA quantities also ensure they will likely equally perform in the fragment library preparation. And of course, as the manual instructs, after the enrichment PCR, the pools are quantified by qPCR and diluted accordingly for flow cell loading. We developed a specific pipeline to manipulate this type of libraries, as they require dedicated trimming on the 2 ends, given the presence of linker adapters and so on. And after the removal of duplicates, we experienced a level that was always below the 10% threshold. With this data showed here on the whole panel of 479 samples, we successfully genotyped almost some 70,000 SNPs as they were selected on the panels, plus more than 450,000 SNPs that were discovered and genotyped with the same criteria as applied to the panel SNPs, which were to be genotyped on at least the 50% of samples, and also the minimum coverage to trustfully call homozygous states was set to 6 reads. This is a typical profile of a SPET GBS experiment, where the reverse reads being all stacked up to maximize the coverage to recall a SNP that was chosen on a given panel of interesting polymorphism, while forward reads are scattered across a larger region and they're ready to explore the diversity, novel diversity, and while providing genotyping information on a cross-sample fashion. This dual genotyping discovery paradigm provides a dramatic benefit in this context, since while the SNPs in panel are recalling common alleles as we selected them during the selection process, the discovered sites does not necessarily follow the same distribution. As we can see in these two plots, the allele frequencies from the genotyping information coming from the panel SNPs basically is recalling the same profile as we choose in the panel. But if we look at the allele frequencies coming from the novel discovered SNP sites and also genotyped sites, we can see how we are sampling very rare allele that would hardly be included in any array-based system. So to conclude, this system, as we adopted it, can both genotype target SNP site and also sample and genotype novel diversity. This targeted enrichment system for genotyping purposes is very promising strategy for experiment where it's required to do fine mapping. So let's say to generate genotyping data for a particular region of the genome, let's say an arm of a chromosome, or to deal with very complex genomes where a full genome sequence is not still available and we are just capability to access the transcriptome sequence. Or, as in our case, to perform genome-wide association studies where the linkage decay is very rapid and researcher must be able to tightly explore only the gene space of such a genome. It's a technique that it's still more expensive than traditional GBS or RAD systems, but it's very competitive to array-based solutions. Also, the ability to scale easily on probe synthesis scales is very helpful for design the most cost-effective strategy. And also the utilization of more probes per SNP can avoid some failure, but in our case does not reward the data points cost. However, if users are eager to deal with very important SNP sites, they can easily think about designing more than one probe per SNP site. And here I'm basically done. I want to thank a member from IGA Tech, which collaborated on this project, people from the University of Southampton, which are the principal investigator of this research project in Populus Nigra, Michael Wright and Gail Taylor. This project was funded by the Framework 7 program, European program called WhatBio, which is a research project on drought resistance on important tree species of Europe. And thanks all of you for your attention.
That was excellent. Thanks very much, David. I think our audience now has a great sense of how SPET can not only be used accurately for genotyping SNPs, but also how this sequencing strategy can be applied to other NGS protocols. Before we start the Q&A session, I want to let everyone know this is your final chance to submit your questions for our speakers. We've gotten some really great questions already, but I'm encouraging all of you in the audience to keep them coming. All right, while everyone is submitting their final round of questions, let's begin the Q&A session so that we can try to get to as many questions as possible.
This is John Sterling, Editor-in-Chief of GEN, and I'm going to be handling the Q&A. So my first question is for David. How does the NGS system remove ascertainment bias? Can you please elaborate?
Of course, thanks for the question. So basically, the system being based on sequencing can remove the ascertainment bias as even by targeting some specific polymorphisms that are previously known, being sampled in some germplasm or any known diversity, the possibility to sequence further sites of the genome, let's say 100 to 200 or 300 base pair upstream or downstream of a given polymorphism, will make a researcher able to catalog any other polymorphism, which will make possible to reconstruct all the haplotypes in that given genomic locus. So the system will not be closed on a biallelic universe, but will be open to a multidimensional universe of all the possible haplotypes. Hope this answers.
I'm sorry, go ahead.
No, I'm done.
Okay, so we have another question for you. Is the lab process longer than with traditional hybridization-based systems?
No, actually, based on our experience, we found this procedure much faster than hybridization-based, as you don't need to wait for a long hybridization procedure. Instead, you just need to follow the primer extension procedure, which is the main extra step compared to a traditional DNA library preparation.
And David, is there any issue related to low complexity of sequence bases?
Fortunately not, because NuGen managed to put some random varying length linker adapter on their construct. So even if the probes are limited in the number, the complexity of the sequencing will be always preserved by those linkers that are differing in the length. So low complexity bases will never overlap, so the sequencer will be always capable to make the correct calibration of lasers.
And David, how can the sequencing platform and chemistry impact the performance of this method?
Well, when sequencer will provide the longer reads, they will also likely make this system able to give a more detailed reconstruction of haplotypes by simply exploring more bases. And it's always a trade-off on the read length and the sampling number. So it really depends on the final outcome that the researcher wants. So there may be scenario where one want to prefer few reads but with long sequencing, or in other cases, like in our, where we want to an aggressive genotyping of many data points without much care of the extra information that we can get out of the rest of the sequencing length.
And David, would it be possible to only sequence the reverse read?
Yes and no. So the system itself requires to sequence the forward read because it's also used for the duplication procedure. So basically, the duplication happens by means of those 6 bases, which are random, generated after the barcode index. And that information, coupled with the position of the forward read, provide an unambiguous duplication of mapped reads. However, I would say that without the forward read, user can keep maintaining the duplication capability only by the 6 random bases, and in some cases this can be very cost effective in a very targeted genotyping experiment like ours, but without paying the money for their forward read.
And David, someone asked, could you provide a more detailed explanation of the deduplication removal process?
Yeah, like I said in the previous question, so basically the duplication happens by looking at every forward map read, which is generated by random sonication, so the likelihood of generating reads that are starting their mapping coordinate at the same nucleotide is very low, but it can happen in very deep coverage situations. So in this case, the information is coupled with the random 6 bases provided by the index sequencing, and so given the random 6 bases plus the position of the forward read, the removal of the duplicates is 100% unambiguous.
Thank you, David. Luke, we did not forget about you. Somebody wants to know, what is the typical timeline to deliver a custom targeted panel?
Turnaround times for custom panels are quite short. Typically, once we've settled upon a design and the target regions, we're able to deliver a custom product in about 3 weeks. This process, of course, involves communication between the customer and Nugen's technical support team to ensure that the design itself and all of the aberrations that a customer is interested in interrogating are fully covered. And so oftentimes there can be a little bit of time required in order to ensure that we've got an optimized design. However, as I mentioned, once that design is settled on and agreed upon, our typical turnaround times are about 3 weeks to ship a custom system out to a customer.
And Luke, what is the limitation of the target region capture size?
To date, we've not yet found a limit to the target region. We've worked on a number of custom designs extending to thousands of genes and tens of megabases in target space. And we expect that this can actually be expanded even further, given the specific need of a researcher for a large target region. We see excellent performance across the entire probe set, and the system itself is able to accommodate a tremendous complexity of probes that are designed and incorporated into the system on a custom basis. So we'd encourage anyone with a very large target region to have a discussion and work with us, and we can continue to push the upper limits of the system.
Great, Luke, thank you. And can non-model organisms be used for custom target enrichment?
Yes, we've used a broad range of different organisms. Of course, today you heard about the poplar. We've worked with prokaryotes, eukaryotes. We've worked within mammalian systems, non-mammalian systems, and essentially we can design to anything that has a reference genome to work from. So again, I'd encourage anyone that is working on any organism, model organism or otherwise, to reach out to us, and we'll work with you to provide an optimized design.
And Luke, do you have any examples of compatible automation platforms for high throughput processing?
Right. So as we've learned, this is a process that's amenable to high throughput process. We've designed the system and the chemistry with high throughput in mind, and we have automated this on different platforms to date, along with many of our other offerings. And we have an excellent automation team that works closely with robotics platform providers to help install and optimize scripts for each given instrument. These platforms include the Cyclone from PerkinElmer, Hamilton platforms, the Bravo from Agilent, just to name a few, but it certainly extends to any programmable open robotics platform.
Okay, thank you very much, Luke. But unfortunately, we have run out of time. But please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends. Thanks again to the panel for outstanding presentations, and thank you to our audience for your attention and for your very thoughtful questions brought up during the webinar. And thank you to Nugent, whose support made this webinar possible. Bye-bye for now.